Om Tham Dhanvantare Namaha. The eye and ear histology. The cornea, microscopic anatomy. The cornea is the transparent avascular anterior one-sixth of eye's fibrous tunic. Histologically, the human cornea is composed of five distinct structural layers, with some modern texts recognizing a sixth transitional zone called Duos layer. Corneal epithelial tissue. Type: non-keratinized stratified squamous epithelium. Thickness: roughly 5 to 7 cell layers deep, accounting for about 10% of total corneal thickness. Cell types: features a single basal layer of mitotic columnar cells, 2 to 3 layers of wing cells, and superficial squamous cells. Key features: superficial cells have microvilli that anchor the tear film. It contains a dense network of sensory-free nerve endings derived from the trigeminal nerve. It has a rapid regeneration cycle of roughly 7 days, populated by stem cells located at the limbus. 2. Bowman's layer. Anterior limiting lamina. Structure: a uniform acellular and non-degenerative sheet. Composition: primarily randomized type 1 collagen fibrils, laminin, and proteoglycans. Function: acts as a tough protective barrier for the underlying stroma. If injured, it does not regenerate and will heal via scarring. Corneal stroma or substantia propria. Volume: the thickest layer, constituting approximately 90% of the entire corneal depth. Structure: composed of 200 to 250 parallel sheets called lamellae. Within each lamella, type 1 collagen fibers run perfectly uniform and parallel. Alternating layers run at orthogonal angles to one another. Cellular component. Flattened quiescent fibroblasts, known as keratocytes, are distributed between the lamellae to maintain the extracellular matrix. Transparency mechanism. Clarity is preserved by the precise uniform spatial arrangement of the collagen fibers and a highly controlled state of relative dehydration. Quote, these decimates membrane, posterior limiting lamina. Structure: a true thick basement membrane that stains strongly with periodic acid stiff PAS. Composition: composed of type 4 collagen and laminin secreted continuously by the underlying endothelial cells. Key features: it naturally thickens with age, evolving from a banded appearance in early development to an unbanded posterior matrix later in life. It terminates peripherally at the limbus, forming the Schwabel's ring. 5. Corneal endothelium, posterior epithelium. Tissue type: a single layer of simple squamous to low cuboidal cells. Key features: these cells possess a distinct hexagonal pattern when viewed face on. Function: these cells do not regenerate in humans. They contain highly active metabolic sodium potassium ATPase pumps. These pumps actively draw water out of the stroma and into the anterior chamber, maintaining the specialized dehydration necessary for vision clarity. Damage to this layer causes irreversible stromal edema and blindness. Or next, retina, histology of retina. The retina is a highly organized, multi-layered sensory tissue lining the back of the eye, structurally divided into 10 distinct histological layers. Functionally, it converts light energy into electrical signals via photoreceptors, processes these signals through intermediate neurons, and transmits them to the brain through the optic nerve. The tissue is oriented from outer, closest to the choroid or sclera, to inner, closest to the vitreous humor. Paradoxically, light must pass through most of the inner layers to reach the photoreceptors at the back. The 10 layers of the retina, outer to inner. 1. Retinal pigment epithelium. Single layer of cuboidal epithelial cells sitting on Bruch's membrane, packed with melanin granules that absorb scattered light and prevent internal reflections. Functions in phagocytosis of shed photoreceptor outer segments and stores vitamin A. 2. Photoreceptor layer, that is layer of rods and cones, contains the photosensitive outer and inner segments of rods and cones. Rods are numerous, highly sensitive, and manage low light scotopic peripheral vision. Cones are dense in the fovea, managing high acuity color, photopic central vision. 3. External limiting membrane. Not a true membrane, but a fenestrated row of intercellular zonulae, adherinates junctions, anchors the metabolic process of Muller glial cells to the photoreceptor cell bodies. Outer nuclear layer, dense sheet containing the nuclei and cell bodies of rods and cones. Histologically, rod nuclei appear smaller, more rounded, and darker than core nuclei. 5. Outer plexiform layer, a synapse-rich zone connecting photoreceptor axons with bipolar and horizontal cells. The retina. Lacrimal gland histology. The lacrimal gland is a compound tubuloacinar and exocrine gland. It is divided into lobules by connective tissue septa and consists primarily of serous acinar cells surrounded by myoepithelial cells and an interconnected ductal system. Key histological components. Secretory acini. The functional units of the gland are grape-like clusters of cells acini that surround a central lumen. Serous cells. These cuboidal cells have heavily basophilic dark staining granular cytoplasm packed with secretory vesicles. They secrete the aqueous layer of tears, which contains vital proteins and lysozymes. Mucous cells, unlike the major salivary glands, human lacrimal tissue is predominantly serous, though it contains scattered mucin producing cells. Myoepithelial cells. Basket-like contractile cells wrap around the secretory acini and small ducts. Their contraction helps squeeze and expel tears into the excretory ducts. Duct system. Unlike the parotid gland, the lacrimal gland lacks distinct striated and intercalated ducts. The ductules arise directly from the acini as intralobular ducts, which are lined by simple cuboidal epithelium and merge into larger interlobular ducts lined by simple or pseudostratified columnar epithelium. Supporting elements. Interlobular stroma. The spaces between the acini and lobules contain loose connective tissue, blood vessels, and immune cells, including lymphocytes, plasma cells, and macrophages. These are critical for the mucosal immune response and antibody IgA secretion. Adipose and age-related changes. As part of normal aging, the gland gradually accumulates fatty adipose tissue and focal lymphocytic infiltrates, which can become more prominent, particularly in most postmenopausal women. The left and flat. Inner ear histology. The inner ear, situated within the temporal bones bony labyrinth, consists of a fluid-filled membranous labyrinth. It is divided into two histological components, the cochlea for hearing and the vestibular apparatus for balance. One, the cochlea for hearing. A cross-section of the cochlea reveals three fluid-filled spiral scalae. Scala vestibuli, the superior chamber, filled with perilymph. Scala tympani, the inferior chamber, filled with perilymph, separated from the middle ear by the round window. Scala media, cochlear duct, the central chamber, filled with endolymph. Key microscopic structures. Organ of Corti, located in the scala media, resting on the basement membrane, resting on the basilar membrane. It contains mechanoreceptors hair cells, one inner row, two outer rows, three outer rows, intertwined with intertwined with support cells, diaters, Hansen and pillar cells. Tectorial membrane, a gelatinous acellular membrane overlying the hair cells. Sound vibrations share the hair cells against this membrane to transmit electrical impulses. Stria vascularis, highly vascularized epithelium on the lateral wall of the scala media that secretes endolymph. Spiral ganglia contains the bipolar cell bodies of auditory nerve, cranial nerve 8. 2. The vestibular apparatus for balance, composed of the article, saccule, and three semicircular ducts, all filled with endolymph, maculae in the utricle and saccule. Specialized sensory regions that detect in linear acceleration and gravity. They feature hair cells embedded in a gelatinous otolithic membrane coated with otoliths, tiny calcium carbonate crystals. Shifts in the otoliths trigger nerve impulses. Cristae ampullaris in semicircular ducts. Elevated ridges in the ampullae that detect rotational or angular acceleration. They contain hair cells embedded in a gelatinous cap called the capula, the inner ear. Histology. These are the reference. Today, we have discussed histology of the eye at the ear. Thank you for patient hearing.